Hello, my dear students. Welcome to our next lecture. This one is going to be on blood clotting. So in the last lecture, briefly, I mentioned about platelets and their function in blood clotting. And we briefly talked about fibrinogen and how that's getting converted into fibrinopeptides. So in this one, we are going to talk in too much detail about blood clotting, different stages of blood clotting, why the blood clotting is important, how the body is preventing us from unnecessary blood clotting, and what's happening during pathological blood clotting. So that's what we are going to be talking about. So as I said in the last lecture, so both the last lecture and this one are absolutely critical for your lectures with me next year. So next year I will be talking about thrombotic diseases, heart failure, atherosclerosis, hyperlipidemia, all those things for you guys. So in order to understand those lectures, these basic lectures, both introduction to blood and blood clotting are important. So please try to remember them throughout your program and throughout your life as well, because we are all studying or learning all basics about our own body and our own blood and blood clotting here. Okay. So what are the learning outcomes for this particular lectures? At the end of this lecture and further reading, you should be able to describe the 5 important steps in blood clotting, because that's what mainly we are going to be focusing on today's lecture. And you should be able to understand the importance of platelets and their activation in blood clotting. Then you should be able to demonstrate the differences between extrinsic and intrinsic clotting cascades and describe what is clot attraction and what is clot dissolution, and describe the pathological conditions associated with the blood clotting, because these are important as pharmacy student. Okay, so we're going to talk about specific definition or technical terms that we will be using in this lecture. So first thing, you need to understand what is blood clotting. Okay, the circulatory system, as we discussed again in the last lecture, is highly delicate. Circulatory system means all the blood vessels and the heart. Okay, that's what is called a circulatory system. This is highly delicate, and if there's any damage to the system, it can cause to the blood to leak. Okay, if there's an injury in the blood vessel, immediately the bleeding will occur. So it's really important to maintain our circulatory system intact without any damage. If there is a damage, there should be a mechanism in order to seal the damage and to prevent the bleeding immediately. That's what is really blood clotting. So basically the blood vessels and the blood are working together in order to prevent minor injuries resulting in death due to bleeding, excessive bleeding, particularly through a normal physiological mechanism called blood clotting or hemostasis. So if there's a injury, immediately the blood has to clot maximum within like around 5 to 8 minutes. Then it will seal the area, prevent the excessive bleeding, so we won't lose a lot of blood. So it's really, really important. So that's what is basically blood clotting. Clot the blood, seal the area, prevent the bleeding. So the definitions, so what are the important terms we're going to be talking about here? First one is called hemostasis, which is basically cessation of bleeding, okay, through blood clotting. Hemo means blood. We know that stasis means standing or stopping or hanging around. It's basically preventing the bleeding using blood clot. Hemorrhage means it's a burst or bleeding, excessive bleeding in the affected area. So this is excessive flow of blood. Again, hemo means blood, rage means burst forth. It's a damage, therefore bleeding. So hemostasis is a physiological blood clotting. And if the same blood clotting is happening under pathological conditions or a disease condition, then it is called thrombosis. Okay, you need to remember that both are blood clotting. One is upon injury and it's happening at the injury site to prevent excessive bleeding. That's a physiological response to clotting. Sorry, physiological response to injuries. That's what is called hemostasis. But if the same blood clot is formed unnecessarily within the blood vessel during pathological conditions or disease condition, then it's called thrombosis. Again, we will talk about that later on. Similarly, hemorrhage is also a pathological condition. Excessive bleeding. If the blood is not clotting, it's a continuously bleeding, then it's a hemorrhage. That's also representing a pathological conditions. Okay, so in this lecture, as I said, we are going to be mainly talking about the 5 different stages of blood clotting. So what are the main 5 different stages here? So the stage 1 is called vessel spasm. As soon as the damage occurs, the vessel will constrict in order to prevent the excessive blood flow in the affected region. That's called vessel spasm. Second one is formation of platelet plug or aggregate and platelet adhesion and aggregation. So basically platelet activation and the formation of platelet aggregation is the second stage. Third stage is formation of the insoluble fibrin clot through cascade of coagulation events. So basically the intrinsic or extrinsic pathways of coagulation will be activated, and that will lead to the production of an enzyme called thrombin, and that will stabilize the clot that's formed by the platelets here. And then the next one, the clot, after the formation of clot, the clot can't stay there forever. It has to get retracted in order to promote wound healing. So that's the fourth stage. So basically clot retraction or squeezing out the clot in order to bring the wound edges together to promote wound healing. Finally, the clot cannot stay there forever. It has to get dissolved and completely removed from our body. So that's what is called clot dissolution. So there are 5 stages in blood clotting. First one is vessel spasm. Second one is platelet activation and aggregation. Third one is coagulation cascade activation to form insoluble blood clot. Fourth one is clot attraction, and the fifth one is clot dissolution. Okay, so this is what is basically happening here. So you can see here briefly injury to blood vessels, collagen is getting exposed and platelet get adhesion in the affected area. Then the platelets will get activated and it will aggregate and make the thrombus formation or blood clot formation. And then the fibrin clot formation will trap all the cells and completely seal the area. Okay, so we're going to go through each stage in detail and talk about them. Okay, what is vessel spasm or basically constriction? So the endothelial injury initiates a vessel spasm or a constriction. When there is a damage, the endothelium within the blood vessel will get damaged. I told you in the last lecture, the endothelium will be in the inner lining of the blood vessel. Okay, so if there is a damage, endothelium will sense it immediately and it will cause the problem. So if there is a damage to the endothelium, it will constrict transiently very quickly the blood vessel. So it will shrink the blood vessel in that area in order to reduce the blood flow at the affected area. Okay, because if you got excessive amount of blood flow here, then the blood flow will be quite a lot and therefore bleeding will be quite a lot. That's why it's making the constriction and it will reduce the blood flow for a brief period. This is a transient event and it lasts for less than a minute. It's a very transient. It's only for a quick time. It's not going to be there for a long time. So this is only quick time and therefore it's reducing the blood flow and to prevent the excessive bleeding here. There is a molecule called thromboxane A2. It's produced from platelets and through other mediator released from the damaged site will contribute to this spasm. And that's what is constricting this area to prevent the blood flow. Okay, so that's about vessel spasm. Try to remember this is a transient event. It will last only for less than a minute, not for longer. Okay, the second most important stage is called platelet activation and aggregation. So we know platelets are circulating blood cells. They are the smallest blood cells and they are mainly squeezed towards the edges of the blood vessel when the blood flow is. This is the lumen. All the red cells will be flowing here, white cells will be flowing here. And compared to them, platelets are very small cells. Therefore, they will be circulating towards the edges of the blood vessel, which is actually very good because the platelets are continuously monitoring or surveilling the endothelial cells. So if there is a damage to the endothelial cells, immediately it will sense the damage and therefore initiate the activation. So platelets get bind to collagen at the damaged site. So if there is a damage, the endothelium will get ripped off and the endothelium is there in a single layer. So it's easy to get ripped off. It's not in multiple layer, similar to smooth muscle cells. So when their endothelium rips off, immediately the collagen, which is present in the subendothelial region, will get exposed. So collagen is a powerful activator for platelets. So it will bind to the platelets, which are already circulating very close to the endothelium. So immediately some platelets initially will see the collagen and it will bind to the collagen, and when they bind to the collagen it will get activated. So when the platelets get activated, they will change their shape. Remember the images I showed you on the journal cover. So the resting platelets will be in spherical shape. When they get activated, they will make lots of filopodia, like finger-like projections, and then finally they will make like a completely spread platelets, which will look like a very sticky film. Therefore, more and more platelets can stick to each other. And at the same time, platelets do have two major types of granules. One is alpha granule, another one is dense granule. Alpha granule contains lots of different protein molecules. Dense granules contains lots of non-protein molecules. For example, alpha granules contain proteins like fibronectin, fibrinogen, von Willebrand factor, these type of molecules, p-selectin, and these are important for blood clotting. Dense granules will contain non-proteinous molecules, for example, ADP, ATP, serotonin, this type of molecules. So these initially activated platelets, they made one small layer, single layer of platelets. OK, and this single layer of activated platelets will release all the factors from their granules. And these molecules will attract more platelets and activate those platelets. At the same time, we're going to talk about the clotting cascade later on. The activated platelets will provide a surface for the clotting to carry on. So at the end of the clotting events, clotting cascades, an enzyme called thrombin will be produced in a large quantity. And thrombin and a molecule called ADP coming from platelet granules, and the molecule called thromboxane A2 that's produced and synthesized and released from activated platelets, all of these molecules will be accumulated in large quantity in the affected region. And all those molecules can activate more and more platelets in the circulation. So when they activate more platelets, they will all clump together because they are becoming like a sticky surface, and they will join together to make platelet aggregate or platelet plug. So initially, even though there was a single layer of thin layer of platelets, activated platelets, now they get lots of activated platelets accumulating in this area. So they will plug the area or seal the area as quickly as possible. OK, so you need to remember that. So first, platelets binding to collagen, getting activated, making a monolayer. These activated platelets will change their shape, release their granule contents, particularly a molecule called ADP. And at the same time, the activated platelets will synthesize and release a molecule called thromboxane A2. OK, and thrombin will be produced on the surface of platelets. We'll talk about that in next slide. And all these molecules will activate more platelets and recruit them to the growing clot. And finally, it will seal the area. But this is only a temporary arrangement because this clot can be pinched off very quickly because it's a soluble clot. It's not been stabilized. So this clot should be stabilized in order to provide continuous protection. So that's where the clotting cascades comes in. So remember the different stages of platelet activation because you are likely to get a short answer question here. So again, I showed you this image in the last slide, last lecture. So this is how the resting platelets will look like. They are discoid shape and activated platelets with all the finger-like projections, pseudopodia or pilopodia. And then the blood clot is completely formed with the activated platelets and the fibrin mesh. OK. OK, so what is the negative regulation for platelets? So certainly we don't want the platelets get activated unnecessarily within our blood. So when we are healthy individuals, our blood platelets are being prevented from getting activated unnecessarily. Only during disease conditions they are getting activated unnecessarily. So here the negative regulation is really important to prevent the platelet activation within the circulation. Otherwise, the clot will be formed everywhere in the blood vessel. Then the blood flow will not occur. So it's really important. So the healthy endothelium we talked about, it's a single layer covering the inner lining of the blood vessel. So this healthy endothelium is producing two important molecules, and these two important molecules preventing the platelets from getting unnecessarily activated within the circulation. Okay, what are these two molecules? So the first one is called prostacyclin. It's a lipid molecule produced from these platelets. This molecule will bind to platelets and it prevents its unnecessary activation. Similarly, endothelium will produce a gas called nitric oxide, NO, that will also go to platelets and prevent the unnecessary activation. So the healthy endothelium in healthy individuals producing these two molecules in order to prevent unnecessary activation of platelets in the normal circumstances. This is how we are circulating blood without making blood clot in our body under normal situations. Okay, so next is activation of coagulation cascade. The third stage of blood clotting is activation of coagulation cascade. So coagulation cascade is really important to make the soluble clot. So even though platelets are making temporary clots, it has to be stabilized. If it is not stable, it will not stay there for a long time. If it is not staying there a long time, the bleeding will start. Okay, so the clotting is basically conversion of fluid blood into gelatinous clot in the affected region, not the entire 5 liter of blood. Okay, that will happen only when we are dead. So this is happening, the fluid blood into gelatinous clot at the damaged site. It occurs, the coagulation cascade will occur on the surface of activated platelets, not resting platelets. You need to remember that. Okay, activated platelets are there only at the damaged region. You will not see activated platelets anywhere around in the body. So only on the damaged site, the activated platelets will provide the surface for the coagulation cascade to occur. There are so many cofactors. For example, particularly calcium is important for regulating the coagulation cascade. Okay, when it comes to coagulation cascades, you got two major pathways. One is intrinsic pathway, another one is extrinsic pathway. Okay, these are really important. You need to remember intrinsic pathway and extrinsic pathway. Both the pathways are leading to the conversion of factor 10 into active form of factor 10. Okay, so we do have all coagulation factors in our blood, but they circulate in inactive form, not in active form. Okay, so we circulate coagulation factors all the time in the blood, but in an active form. Only during coagulation or blood clotting, they will be converted from inactive form to active form. Okay, and the activated coagulation factors are the one playing roles in coagulation cascade, not any other molecules, not the inactive molecules. Okay, try to remember that. Okay, so the intrinsic pathway is a relatively slow process. You can see here it's got multiple steps. It starts from factor 12 and going all the way up to factor 10 conversion into active form of factor 10. Extrinsic pathway is quite quick. It's a faster pathway. It's induced by the injury. When the injury happens, immediately at the injury site, tissue factor will be released. This tissue factor will convert factor 7 into activated form of factor 7, and it will go to the next step here, factor 9 into active form of factor 9, and then straight away convert factor 10 into active form of factor 10. So this is the common pathway for both of these pathways. So there are two pathways, intrinsic and extrinsic pathway. Intrinsic pathway is slow pathway. Extrinsic pathway is quite really faster pathway. Intrinsic pathway starts with factor 12. Extrinsic pathway starts with factor 7, activated mainly by tissue factor. And the common pathway for both of them is conversion of factor 10 into active form of factor 10. So try to remember that. Activation of one factor leads to activation of another factor. So this is a cascade event. When you activate one factor, immediately that will go and activate next, next. It's like a signaling cascade. That's why this is called coagulation cascade. So at the end of the coagulation cascade, thrombin will be produced. When the active form of factor 10 is produced, is a common pathway for both the pathways. Prothrombin will be converted into thrombin. Thrombin is a very, very important enzyme in our body. It's a multifunctional enzyme. It can do lots of things. In the previous slide, I mentioned thrombin will activate platelets because it's a powerful activator of platelets. So here what is it doing is basically converting the fibrinogen into fibrinopeptide. So again, we know fibrinogen is an inactive clotting protein present in the plasma all the time. OK, but it will not make the clot. It has to be converted into fibrinopeptide, three different types of fibrinopeptide, alpha, beta, gamma. And these peptides will make the blood clot. That's when the clot will be fully completed. So thrombin is playing a critical role here. At the same time, thrombin is converting the last factor that's called factor 13 into activated form of factor 13. OK, this is really important. So here this fibrinopeptide will cover all the platelets, activated platelets, because platelet plug is already formed. It will cover everything, entrap all the red cells and the white cells, but still it's not really stable. It can get pinched up or broken down. So it's really important to stabilize it. So that's where this active form of factor 13 comes in. This factor will come and cross-link all the fibrinopeptide. So when it's cross-linking the fibrinopeptide, the clot is completely stabilized. So that's when the clot is really jelly and thick, seal the area, and therefore blood will not leak. Okay. So important points here. There are two pathways, intrinsic and extrinsic pathway. Both the pathways and all the coagulation cascades are happening on the surface of activated platelets. And the common pathway here is the conversion of factor 10 into active form of factor 10, which will convert prothrombin into thrombin. Thrombin is an important multifunctional enzyme. Thrombin is activating platelets. Thrombin is converting fibrinogen into fibrinopeptides, and thrombin is converting factor 13 into active form of factor 13. Factor 13 is the one stabilizing the clot by cross-linking the fibrinopeptide. Okay, and various clotting factors or cofactors are associated here. For example, calcium. Okay, so this is the next step important in the clotting cascade. Okay, so we will move on to the next one. So here you can see this is a fibrin. It's a full protein formed, and this is how it will look like. It will look like a filter which will entrap all the cells. OK, so I will talk about the remaining things in the next video. Thank you.